for joining us on TBB channel. My name is Alex, and today we'll speak about a new trend in the scientific field, biotechnology. For 20 years, a lot of companies have invested in this field. However, biotechnology has been used for more than 6,000 years to make useful products such as wine, beer, bread, or even cheese. Just look around me, you can see this wonderful vineyard. The final product, the wine, is one of the oldest biotechnological products, with people involved in this production by using microorganisms such as yeast. Biotechnology combines cellular and biomolecular processes to develop products to improve our life and the health of our planet. Now, let's talk about the history of this field.
What is amazing about biotechnology is that even if we have used it for a while, we had to wait for the 17th century to understand what a microorganism was. A century later, the discovery of DNA and proteins opened the gate to life science. However, we had to wait another century to get how DNA was organized. Since then, scientists have started thinking about how to use this knowledge to modify microorganisms to develop biotechnological products.
Its applications are everywhere around us. In seed modification to improve the crop yields, milk production optimization by selecting specific cow species, yogurt development with new properties, or even soil decontamination with bacteria. But the biggest part of this market is the pharmaceutical industry with the development of new specific drugs. The most common biotechnological production concern recombinant proteins such as insulin or antibodies.
With the potential of the biotechnology market, some schools have specialized in this area.
Right now, I'm standing in front of the ENSTBB, a biotechnology school in Bordeaux that trains students in the characterization, production, and purification of biomolecules. Let's talk with Dr. Djirik, who is specialized in molecular biology. So, Dr. Djirik, can you tell us why it is important to study biotechnology nowadays?
To my mind, the big pharma companies are now moving their process to biotech instead of chemical process. Here at the ENSTBB, the education allows students to learn every steps to properly set up a biotechnological process in the pharmaceutical field.
In order to help you to understand what is done in this school, let's see the different steps of a biotechnological process.
The first step in a biotech process is molecular biology. Once we have chosen the specific protein we want to produce, its gene sequence is generated. The vector, a carrier of DNA, is cut by restriction enzymes to insert the gene. Ligases are then used to stick the gene onto this vector. The plasmid is then transferred into a bacteria who will produce our protein. Bacterial colonies will grow on a petri dish.
Depending on the protein you want to produce, the host is chosen for bench or large-scale productions in bioreactors like this one. This is the culture step, also called upstream process. For instance, mammalian cells, bacteria, or yeasts can be used to produce virus or recombinant proteins. Once a product of interest has been produced, it is necessary to separate it from all the other molecules. Specific resins are packed into this kind of column to purify proteins. Molecules will be specifically coded by the resin due to electrochemical and affinity interactions, whereas the other compounds will go through directly into the waste. Now, you have all the elements to understand what biotechnology is, and I hope it convinces you how it is a promising market.